Hi guys, it turns out we do need to know how to do a dihybrid cross for the test. So we're going to start with that for exam 2. And then we will do chapter 21, Patterns of Inheritance, part 2. So here we go. Alright, Punnett Square. So in the last lesson, we looked at monohybrid crosses, which are just looking at one gene, but we can also do dihybrid crosses where we look at 2 different genes. So the genes we're looking at on this one are the presence of freckles and then finger length. So the allele for freckles is, I just assigned it an F, so it's dominant with a capital F, and then freckles, no freckles would be the allele would be a little f. And as for finger length, why is it D? Because now I'm going to have to say big D, and that makes me uncomfortable. Big D is for long fingers, and little d is for short fingers. So those are the different alleles. And long fingers are dominant to short fingers. So I know this looks scary, but it's actually pretty simple. So it says, what is the phenotypic ratio of a cross between 2 heterozygotes? Remember, hetero means different, for both freckles and finger length. So if we write out the genotypes for each of those, I feel like I'm not getting good light here. The parent genotypes would be, running out of space here. Big F, little f, big D, little d. Try to put a little tail on my little f so you can see. And we're going to cross that with someone with the same genotype. They're both heterozygotes. Heterozygous, what am I trying to say? So this is simple. So basically what you do is you got to find out the different allele combinations that they could make, the different genotype combinations that they could make. So this person could make a big F and a big D when they're making their gametes. So think of these as like, they're splitting up to make the gametes. It could also make a big F, little d. And it could make a little f, big D, and then a little f, little d. Did I do that right? Is that all the possible combinations? Those two, those two, those two, and those two. Okay, and then this one's going to be the same because they're both heterozygous. So what you do now is you just bring these gametes down here. So that's one parent, the possible gametes they can make, and this parent can make the same, so we'll put the same ones down the side. Okay, and then it's just the same thing with the monohybrid crosses. And remember to always do the capital letters first, and I've just decided to do the F's first, so we're always going to keep the F's together, and then do the D's second. So this would, these gametes fertilize each other, you would get big F, big D, big D. Now let's drag these down and over. This would be big F, big D, little d, if these gametes came together. This would be big F, little f, big D, big D. and then big F, little f, big D, little d. And I'm just going to go ahead and fill the rest of that in. Alright, so I did that pretty quick. I'm trying to figure out what's causing this shadow here. Is it me? So if you need to pause it for a second and get that filled out, you can. And then down here, you'll need to give the genotypic ratio, phenotypic ratio, and the phenotypes produced. So the possible genotypes, we've got big F, big F, big D, big D. We have big F, big F, big D, little d. Got big F, little f, big D, and big F, little f, big D, little d. And let's see what else we're missing. And I'm just getting this from this chart here. We can have big F, little d, little d. I'm gonna have to take up 2 rows here. Did I get them all? Little f, big D, big D. Sorry, this takes a minute to think through here. Little f, big D, little d. Big F, little d, little d. And then I think little f, little d, little d. Alright, so we can just go ahead and count these up. How many big F, big D, big D's do we have? Looks like we have 1. Big F, big D, little d. We've got 1, 2, 'Big F, big D. 1, 2, 2 on that one. Big F, big D, little d. So the heterozygote, like the parents. 1, 2, 3, 4. E. 1. Just 1. Little f, big D. 1, little f, big D, little d. One, see which one. Two, and then just one of these. So let me make sure those all add up to 16. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. Did I miss this one? Well, based on the math, I can tell it's going to be 2. Let's double check that. 2 little f's. Yeah. Okay. Wow, there we go. So, what this will come out to, sorry, I'm adjusting something on my computer. Nope, not that. Oh, that's why it looked dark. My computer screen was way, way dimmed. Okay, we're good. All right, so you could write it out like that, or you could say 1 to 2 to 2 to 4 to 1 to 1 to 2 to 2 to 1. And just so you know, on the assessment, if you have to do a dihybrid cross, and you will, it's not going to be 2 heterozygotes, which means that the genotypic ratio won't be so messy. Okay? All right, phenotypic ratio. So let's write out the different options that we can have. There can be freckles with long fingers. We can have freckles and short fingers. I'm just going to write short. We could have no freckles and long fingers. We could have no freckles and short fingers. And we could have no freckles and short fingers. Okay, so I'm not doing anything magical here. I'm just looking at the different alleles that we have and how they could possibly combine. So for the phenotypic ratio, let's first count up freckles and long fingers. So we know that if they're going to have freckles, and I might have done this backwards from the paper. I think no freckles was the dominant gene. Okay, so if they're going to have freckles, they need just one big F. And if they're going to have long fingers, they need at least one big D. So let's find these and mark them with a circle. You can mark them however you want, but anything that has a big F and a big D, I'm going to put a little circle here in the corner. Okay, so all of these on the top row did. All right, so let's count up our circles. We have 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, now let's look at freckles with short fingers. And since short is recessive, in order to have short fingers, they need 2 little D's. And to have freckles, they need at least one big F. So I'm just going to go look through 2 little D's. I'm going to mark these with a square. Okay, so that's 1, 2, 3. That would have freckles and short fingers. And now let's look for no freckles and long fingers. So no freckles would need to be little f, and long fingers need at least one big D. So little f, where are you? This one would. Let's mark it with a triangle. That one would. And so would that one. So we count up our triangles. That's 1, 2, 3. Why did I do new? I did this one twice. Okay, and then this last one here, we'll give it a heart, is no freckles and short fingers, because you would need two. It would need to be homozygous recessive for both of the genotypes. So that's just one. So we would state this phenotypic ratio. You could write it out like that, or you could say 9 to 3 to 3 to 1. 9, 3, 3, 1. Okay, that took longer than I thought. We're 15 minutes in. I'll talk about some extra practice on these. Actually, I'll do it now. On Canvas, there is a Punnett Square practice that I think it says it's optional. So it has a bunch of problems, and you can go through and try to solve those. And then Amber Mortensen, she's great. She's made like an answer key and an explanation video to go with that. And those are both on Canvas under Module 2. So if you need some extra practice, then I would recommend it. Go do that assignment. And again, it's not graded or anything. It's just for extra practice. But you will need to know how to do a monohybrid cross for the exam, a dihybrid cross, and then list the genotypic ratios as well as the phenotypic ratios. Okay, moving on. Our question of the day here is, can inherited color blindness be treated? And if I have this printed in color, you could see what normal vision was like compared to red-green, compared to blue-yellow color blindness, but mine all looks the same, so sorry about that. My color printer finally came, so I just gotta get that set up now. So for the pre-lecture assignment, this top part, I'm gonna skip because we're going a little slow here. But the bottom part, I do want to go over. Actually, you know what? We're gonna skip on to the next page. Sorry, I'm all over the place. I just got home from work with all the 12-year-olds, so I'm a little scatterbrained. Trying to get it together here for this. Alright, so genetic disorder. What it is, it's inheritance of altered or defective genes responsible for many human diseases. So family history and pedigree charts can help identify disease probability. Whoa, sorry for my tiny handwriting. Zoom that in for a second. Family history and pedigree charts help identify disease probability. Alright, so what a carrier is, we're going to use the word carrier a lot today. It's an individual who carries the allele for a recessive trait but does not express it because it is paired with a dominant allele. So, out of the three types of genotypes we've learned. There's homozygous dominant, homozygous recessive, and heterozygous. Which of those would be carriers? Well, it's going to be the heterozygotes because they have one of each allele. So heterozygotes are carriers, so they don't have the disease, but they carry the allele for it that they could possibly pass on to their offspring. You guys, I'm sorry. Okay, so let's look at how these pedigree charts work. So anytime you see a horizontal line connecting two individuals, that shows a marriage or a mating. I'm just going to say marriage. And the vertical line down, if it's connected to that, shows a child. So here we have two more individuals that are married with one child, and the squares represent the males and circles represent the females. And then if they're shaded in, that means they are affected. They have the whatever disease you're showing in your pedigree chart. Okay, autosomal recessive patterns of inheritance. So there are autosomal recessive disorders and there are autosomal dominant disorders. So with autosomal recessive genotypes, to be affected, you have to have both of those recessive alleles. So the affected genotypes would be two lowercase letters. We'll use A again. So little a. And unaffected, if you have a dominant allele at all, then it's going to mask that recessive allele. So unaffected could be big A, just homozygous dominant, or it could be heterozygous. So they're not affected, but they are a carrier for whatever recessive disease we're talking about. For autosomal dominant disorders, the unaffected genotypes would be little a, because if you have that big A, you are going to be affected. You're going to have the disorder. So the affected genotypes is anything with a big A. And let's look at this a little more in detail here. I'm not going to talk about the genotypes necessarily, but just the patterns of marriages and offspring. So this right here would represent a marriage between these two, and then they had these two offspring here. So you might be tempted to think that these are their offspring as well, but these are people from the outside that came in and married their two children. Their two daughters, it looks like. So again, the horizontal line represents a marriage, and then this couple here had 3 offspring, and this couple had 1 offspring, and oh, something weird happened. So these two would be first cousins, right? And is it recommended to marry your first cousins? No, it's not. I must say, I am Swiss. My heritage goes way back in Santa Clara here, if you know where that is. And so I don't know if it's because the pickings were slim or what, but my grandpa's parents, so my great-grandparents were first cousins. And anytime something's wrong with me, I just go to that for an excuse. And the reason why that's not recommended, you'll see here, these 2 cousins married and had 3 children, and what ended up happening is they were both carriers of that, what am I looking for? That affected allele. And so when you have relatives that marry each other, they're most likely to carry the same recessive alleles that can code for these disorders. And then look, 2 of their kids were affected. All right, so don't marry your cousins. And you're going to be shown, oh, let's read this last part here. An affected individual will have inherited the dominant allele from an affected parent. So you'll be shown these pedigree charts and then expected to decide if it's showing an autosomal recessive or autosomal dominant gene or disorder. So hints for determining autosomal recessive: 2 unaffected parents can have affected offspring if both are heterozygotes. So if you have a big A, little a, and a big A, little a, both of these parents are unaffected because the disease is recessive, but they can both pass on their little a's to have an individual offspring that is affected. And then if both parents are affected, they can only have affected children. So if you have 2 affected parents that mate, the only alleles they can pass on are little a's. And so 100% of their kids are going to be little a, little a. Affected individuals can skip generations. And with these autosomal recessive, not very many individuals are affected in total. Hints for determining if it's an autosomal dominant disorder: 2 affected parents can have unaffected children if they're both heterozygotes. So again, to have the disorder with an autosomal dominant disease, you would have to have at least 1 big A. And if they're both heterozygous, they can still pass on each a little a to their offspring, and then their offspring could be unaffected. It usually does not skip generations, and there are usually more individuals affected in general. So here's an example of an autosomal dominant pedigree chart, meaning the ones that are shaded in have an autosomal dominant disorder. So we have 2 parents that are both affected because they each have a big A. And then these are their 4 offspring. You can see their phenotypes. And to do this, to see if it's possible, well, we'll get there later. We can't really determine all the time if someone is shaded in, if they're big A, or big A, little a. We just know they have at least 1 big A. If they're not shaded in, we know they're not affected, so we know that they're all going to be little a, little a's. So let's move along here, do some practice of this. So you'll be shown examples like this. So what is the genotype for each individual? So it wants us to say whether each of these are big A, little a, or little a, little a. And remember, the squares are males, the circles are females, horizontal line represents a marriage, and then those connected are the offspring. So let me bring our paper back here. Just looking at this one right away, and what you can do, and this is actually how I used to teach it for just the general biology course, is if you don't understand these well, like I can look at this and see whether or not it's autosomal dominant or recessive just because I've done so many, and maybe your brain can work that out as well. But if it doesn't, use a pencil and just randomly guess if you think it's autosomal dominant or autosomal recessive. This will all make sense in a minute. And what is the inheritance pattern shown for each pedigree? So it's asking, is it autosomal dominant or autosomal recessive? So just take a minute and look at this. We'll look at our tips. Let's see if we think it's autosomal recessive. So 2 unaffected parents can have affected offspring. So we do have 2 unaffected parents that have affected offspring. If both parents are affected, they can only have affected children. We don't have a case where both parents are affected here. Affected individuals can skip generations. That's what happened here. They skipped a generation. And not many individuals are affected in total. So 2 out of 1, 2, 3, 4, 5, 6, 7, 8, 9. I mean, it's not a huge amount. So let's just assume here that this is autosomal dominant. Or, excuse me, autosomal recessive. Just based on those hints. So we're going to test it out. So what that means is that any individual that is shaded in has to be little a, because they have to have both recessive alleles in order to be affected. Now for this, we know there's a big A here somewhere, but can we say for sure whether or not it's big A, or big A, little a? Well, it could be either. We can't say for sure. So we're just going to leave it as a dash here. Or and let's just go ahead and add big A's to all of these because we know they have at least one. To not be affected. Just thinking how I want to work through this here. Okay, let's focus on this little family right here first. So they do have one affected child. So that means the parents have to be heterozygotes in order to pass on that little a, 2 little a's to the offspring. And in that case, let's put them in a Punnett square. So just splitting up the alleles. Okay, so they can have that little a, which you see here. And they can have, their offspring can have big A, or big A, little a as well. So we don't know what these are. So we kind of draw a dash like that. We just know they have a big A somewhere. All right, and the same goes if we're looking at this family now. This one also could be big A, or big A, little a. So let's do a Punnett square of each of those. So we know one is little a, one could be big A. And 100% of those would be big A, little a. If this was a big A right here. And is that possible? Yeah, because none of them are affected. They all have a big A, and one of them we know has a little a in order to pass that to their offspring. So let's try the other possibility, big A, little a, for this one. All right, would that one be possible too? And you might be tempted to say no because of these little a's here. But they could have still had all 3 kids be big A, little a. So yeah, we can't say what that one is, which means that we also can't really say what these ones are. All right, so this is an autosomal recessive inheritance pattern. So the individuals with 2 of the recessive alleles are affected with this disorder, and then this is as far as we can get with the other individuals. Okay, let's try this second one. And I'm going to just assume, since the first one is showing autosomal recessive, that maybe this next one is showing autosomal dominant. So let's look at our hints. 2 affected parents can have unaffected children if they're both heterozygotes. So we do have 2 affected parents here. and they have unaffected children. So there's a hint that this is autosomal dominant. The next hint is that it usually does not skip generations. Look at that. We have it in all 3 generations shown here. And there are more individuals affected in general. And here we've got 1, 2, 3, 4, 5 out of 1, 2, 3, 4, 5, 6, 7, 8, 9. So that percentage is much higher. So let's just assume this is autosomal dominant. And then we'll fill everything in and see if it works. Okay, so if it's autosomal dominant, we know that all of these unaffected individuals are homozygous recessive. They don't have any big A's, because if they did, they would be shaded in. So we can say these are all little a, little a. And we know that all of these have at least one big A because they are affected. Alright, if we look at this first marriage here, they have an offspring that's little a, unaffected. And the only way that that could happen is if this parent had a little a. So we can say that. Let's throw it into a Punnett Square to test it out. So big A, little a, little a, little a. Alright, so they can only have children that are little a, or big A, little a. So we got our little a here. None of these can be big A. That's not one of our possibilities. So we can go ahead and say they're all big A, little a. Okay, now these 2 affected individuals mated. And it looks like, let's do a Punnett Square for this generation as well. Big A, little a, little a. Alright, so they can have homozygous dominant, they can be heterozygous, and they can have homozygous recessive, which you can see here and here. And so can we say for sure whether this is big A, or big A, little a? No, we can't, so we'll just leave it as a dash. So the pattern of inheritance here, it does work. It's autosomal dominant, and that's the genotypes we can come up with for each individual. So I would show an example of what would happen if I started off wrong, like assuming that this was autosomal recessive, which is fine as long as you get the right answer ultimately. But if this was autosomal recessive, let's just do it on the top here and see. If that's what we assumed originally, that means all of these would have to be little a, and you don't have to fill this in. I'm just showing you guys. Okay, and these people would have to have a big A. So could this, if this was big A, little a, oops, I'm confusing myself now. Yes, so to start with, it could form, oh, but we don't know what these are. Okay, so this could be, this would have to be a big A, little a. Okay, and then we have little a, and a little a here. Could they have any offspring with big A's like you see down here? No, they could not. So now we know this is not autosomal recessive. Autosomal recessive, and that it's autosomal dominant. If that completely confused you, forget about that second demonstration I just did on that pedigree chart. But yeah, I would try to remember the hints for each one, and then just look at it and make your best guess of which one you think it would be between autosomal recessive or autosomal dominant, and then start filling it in. And if something doesn't match up, erase it and switch it to the other one and try again. Okay, let's make it even more complicating. Just kidding, it's not too bad. So codominance. This is when two alleles are dominant in heterozygotes. So you can have, and let's take an example that is not real, but let's say that you have two possible alleles for flower colors. One for one's an allele for red petals, and one is an allele for pink petals, and they have codominance, so they're both dominant. So in the offspring, you would expect to see some parts of their petals red, and some parts of their petals white. So a real example of this in humans is blood type. So AB blood type. So in this case, both A and B are dominant, and both are expressed in the phenotype. Okay, so A and B is just referring to a type of antigen that you have on your red blood cells. And the blood types, if you don't know, are either, and we're not going to bring positives and negatives into this today. There's A, B, AB, and O. Those are the four possible blood types. So, if you get an A allele and a B allele, you're going to have A and B antigens on your red blood cells. You'll have both. So codominance, both of the dominant alleles are expressed. So we know this guy here, the O allele is recessive. So let's list out those blood types. If you are type A, your possible genotypes could be big A, or big A, O, because the A is dominant to the O. If your blood type is B, you could be big B, or BO. Oh, who wants to be BO? It's okay. If your blood type is AB, then the only genotype that you could have is AB. And you're exhibiting codominance because you're showing both alleles. You're expressing both alleles. And then for type O, your only genotype can be OO. So if we look at this, if we have parents that are type A and type B, what possible offspring could they have? Well, and we don't really know if this is an A or a B right here in these spots until you look at their offspring. If they have offspring with type O blood, then you know that they are both carriers for that recessive O. So anyway, we plug that into our Punnett squares. There's our eggs. Here's our sperm. We split each of these up, made some gametes, made a Punnett square. And so these type A and B parents could have all 4 possible blood types. So their offspring could be AB, they could be B, they could be type A, and they could be type O. Alright, multiple allele inheritance. A gene can have several possible alleles instead of just 2. So far we've only looked at it with just 2 possible alleles. But each person can have only 2 of the possible alleles. Okay, so there are multiple allele types, but you only inherit 2. So an example again is ABO blood types. So instead of just having 3 or 2 possible alleles that you can inherit, there are 3 different possible ones. Okay, so A, that's a B. ABO blood types, 3 allele options, but you only can inherit 2. Alright, let's get into X-linked inheritance. So these are genes located on the X sex chromosome. Remember, all humans have 1 pair of sex chromosomes, and biological females are XX and biological males are XY. And so this X-linked inherited traits this is actually really interesting. They're only located on the X sex chromosomes. So they can be either dominant or recessive. And so an example of this is red-green color blindness. So this is a recessive trait that's carried on the X chromosome. And more males are affected than females with these X-linked inheritance disorders. And so the question is why? So if you think about it, X-linked inheritance almost always requires a couple visual examples to show why men are more affected. So we'll look at that in just a minute. And men only have 1 X chromosome, so if that one is bad, then there isn't another one to mask it. So in order for a female to get an X-linked recessive disorder, she would have to get a recessive X from mom and a recessive X carrying from dad. For the males, they only have to get it on 1 of their 2 sex chromosomes. Okay, so let me summarize that a little better. Men only have 1 X chromosome. So if it's bad, there isn't another to mask it. So let's look at this Punnett Square here. And with sex-linked disorders, we usually draw the X's and Y's, and then we attach the alleles to whatever chromosome they go with. So here we have, let's see, X big B Y with X big B X little b. So again, we just break down those alleles, X big B, Y. Here would be X big B, X little b, and then same thing, you just drag them down and over. So X little b, X little b. This person would be unaffected, and these are two unaffected parents, by the way. This one would be X big B, X little b, so they wouldn't be affected. This one, X big B, Y, wouldn't be affected. But this poor guy happened to get the X little b, Y, so they would be affected by whatever X-linked trait we're talking about, say color blindness, for example. That's why you find more men that are red-green colorblind than females, because they only have to inherit it on that one X chromosome that they have. Alright, some other X-linked inheritance examples are hemophilia. I think we might have talked about this earlier, but this is when you have defective clotting factors in the blood. And so these people can easily die from a minor injury because their blood can't clot and they bleed out and get lots of bruising and can have lots of problems. So another is Duchenne, I think is how you say that, muscular dystrophy. It's a specific type of muscular dystrophy, and this is where your muscle fibers degrade. And the prognosis for this one is typically not very good because what will eventually happen is the muscles that help you breathe will start to degrade, and then you can no longer breathe on your own, and then that is not a good thing. So if we look over here at this pedigree chart, can a female ever be affected by these, and how? So again, the ones that are filled in here are affected. So this is showing sex-linked or X-linked recessive because all of those that are filled in have no big Bs. So let's say you have a mom who is X big B, X big B, so she's not a carrier, but she's married to, I'm going to just use color blindness again as an example, a colorblind man. He's X little by. So their offspring happen to be X big by, so an unaffected, oh no, that's not their offspring. Their offspring are, their daughter is going to get, all of their offspring are going to inherit this affected X chromosome from their dad. The daughter got, let's see, X big B, X little b, so she's a carrier, and the son, is X big B, Y. So he happened to get the unaffected from mom. Sorry, I'm being confusing. And then each of those siblings had a marriage here, and this male was X big B, Y, so not affected, and they had 4 offspring, and one of those was affected because it got this mutated X chromosome from mom. This one's a carrier, this one's not a carrier, this one's unaffected. And then this one over here married a colorblind female, which is rare. But in order for that to happen, you would have to have a colorblind dad and either a colorblind mom or a mom that was a carrier. And their offspring son was also colorblind. So I'm kind of going out of the way here, but let's show how a female could be affected. So if dad is affected, it would be X big by, and if mom is a carrier, let's see if that can get us an affected female. So we got X big B. I didn't make this big enough. Or X little b. Already we've got an affected female. So yeah, if dad is colorblind and mom is colorblind, they can have a colorblind female. Let's try the other scenario where mom is just a carrier and dad is colorblind. Zoom you way out here. Okay, so dad is colorblind, so he's got that little b on his X chromosome. And mom is not colorblind, but she's just a carrier. Let's see if they can make any females that are colorblind. So X big B, X little b. That's a female, not colorblind, but a carrier. X big B, Y. That's a male, not colorblind. X big B, X little b, X little b. So that is a colorblind female. And X little b, Y. That's a colorblind male. So, yes, a female could be affected if they inherit two affected X's. So that's if dad is colorblind. Dad has to be colorblind. And then mom can be colorblind or heterozygous, a carrier. Okay, so this is X-linked inheritance examples continued. We just looked at color blindness. That's what we talked about before, but this is where you have defective cones, and so you can't see all the colors clearly because the cones are the color sensing sensory cells in the retina. So back to the question of the day, can inherited color blindness be treated? So let's think about this. Color blindness cannot be cured, but they do have contacts and glasses that are colored to help make colors more distinguishable. I don't know if you've seen the TikToks of those videos or YouTube videos of someone that's colorblind and they're given these glasses that just helps the colors be more distinguished. To be able to see the difference more clearly, and their minds are just like blown, and I think it's so cool. Sometimes cones are defected and overlap, and these glasses and contacts help lessen that overlap. And if colorblindness was acquired later in life, they can also help, but usually they try to treat the underlying cause first. So in short, can inherited colorblindness be treated? No. But you can get some fancy glasses or contacts to help distinguish colors. So that would be a temporary fix, not a cure. Okay, so here are some blood typing color blindness practice problems. I'm not going to do these. I think there is a solutions video on Canvas, but you can try this out. So, and they actually can use this in some cases. So, a woman with type A blood is claiming that a man with type AB blood is the father of her child who is type O blood. Could this man be the father? Can we rule him out as a father? You know what? I want to do this. Let's do it. So the woman is type A blood, so she can be either A, or AO, but we know she has a type O child, so she's got to be AO. And she's claiming this AB guy is the father of her kid. So let's put these guys in a Punnett square together and see if they could have a type O child. Can they? No, they cannot. So she's wrong. She got her dates wrong, or she's lying. But we can definitely say he is not the father. No. The next one, a man with AB type blood is married to a woman with type O blood, and they have 2 natural children and 1 adopted child. The children's blood types are A, B, and O. So which child is adopted? So we know with AB blood type, the only possible genotype is AB. And with O blood type, the only possible blood type is OO. So let's throw that into a Punnett square here. These are my favorite to do, I'm sorry. So AB, OO. So we can have AO, BO, AO, BO. So they can have children with blood type A and blood type B, and that's it. So the adopted child is type O here. Okay, what are the odds a woman with a genotype X big B X little b and a man with a genotype X little b Y will have a colorblind son? Remember, colorblindness is recessive. So we'll say big B is normal vision and little b is colorblind. So let's Punnett square this up again. So we've got an X big B and X little b woman. My B's are getting sloppy. And we've got, so she's got normal vision, but she's a carrier for color blindness. And we've got a colorblind male. So the chances they will have a colorblind son. So this would be X big B, X little b. So that is female that has normal vision, but is a carrier. X. Sorry, I was taught to write them on the top. I'm sorry if that confuses you. Just have it. X big B, X, or X little b, X little b. Ooh, look, we got a colorblind female. This one, X big B, Y. That's a normal vision male. And X little b, Y. That is a colorblind male. So it's only asking, and make sure you look closely at this, to have a colorblind son. So we're only looking at the XYs here, and that's a 1 out of 2, or a 50% chance, or a 1-to-1 ratio. Okay, I'll let you guys try that last one on your own. And then for this last page here, these are pedigree chart practice problems with solution videos on Canvas. I need to verify that those are still on there. Again, they were made by Amber Mortensen. And so you can try these out. And what you're trying to figure out is if each of these are showing autosomal dominant or autosomal recessive, and then the possible genotype for each individual. So look at the hints you were given, and then you can randomly pick one and guess. And if you were to guess that this was autosomal recessive, then you know that the two shaded in individuals have to be little a. And then you can see if that works out. If you were going to assume that they were both autosomal dominant, these two shaded ones would have to have at least one big A, and all the rest of these unshaded would be little a. And then you'd have to maybe do some Punnett squares to see if that would work out. So you can practice those, and then the answers, there should be a video on Canvas for that. So sorry this went a little bit long, and I'm even starting to lose my voice from talking too much. And I feel like I was kind of all over the place today, so I apologize. If you have questions, please let me know, and I hope you guys have a great day.